Good morning, good afternoon, and good evening, everyone. I'm Ron Vale from Janelia Research Campus, and I'd like to welcome everyone to Life Science Across the Globe. So we're now in our third round of this seminar series of 8 sister institutes, and we're back to Janelia Research Campus, and then we'll be traveling around the globe in the next few weeks to other sister institutes. Today, I would have the great pleasure to introduce Janine Stevens, who will introduce the speakers from Janelia. Janine is the main organizer of life science across the globe. She received her PhD from Drexel University and then did a postdoctoral fellowship at the NIH. Janine then joined Janelia in 2008, and she's been the director of scientific programs and is responsible for a large number of conferences that we run here at Janelia. Of course, now we can't hold those conferences in person, and Janine has very skillfully pivoted to organizing quite a number of online events, including Life Science Across the Globe, of which she's the main organizer. And this involves an enormous amount of work and coordination with all of our sister institutes. And I'd like to personally thank her and recognize her accomplishment in making this series run so smoothly and so successfully. So Janine.
Thank you, Ron, for that very nice introduction. Hi, everyone. Thank you so much for joining us today. I am very excited to welcome you all back to Janelia for this week's talks, and I am especially excited to be introducing both of our speakers today. So our first speaker is Kristin Branson. Kristin completed her undergraduate work in computer science at Harvard University and did her graduate research in machine vision and learning at UC San Diego with Serge Belanger and Sanjoy Dasgupta. She completed her postdoctoral work at Caltech with Pietro Perona and Michael Dickinson, developing computer vision software for automated tracking and behavior analysis of flies. Kristin joined Janelia as a lab head in 2010, where her lab continues to develop tools for quantitative animal behavior analysis. She is currently a senior group leader at Janelia, as well as the head of our Computation and Theory group, which she will tell us more about in her talk today. I also want to mention that Kristin and her lab are very passionate about making science, and particularly computer science, both exciting and approachable for kids. And they have started and contributed to several different science outreach programs at Janelia. Our second speaker today for our culture talk is Tang-Liang Chu. Liang studied biochemistry at the University of Wisconsin-Madison and completed his PhD at St. Louis University, studying myosin regulation in endothelial cells. Interested in addressing both the spatial and temporal regulation of signaling pathways inside of cells, Liang went on to do his postdoctoral work with Rex Chisholm at Northwestern, developing a fluorescent biosensor to simultaneously monitor enzymatic activity and localization of myosin light chain kinase in vivo. Liang then went on to become the director of the Center for Advanced Microscopy at Northwestern's Feinberg School of Medicine, and then director for University Imaging Resources, leading the overarching strategy to build integrated imaging infrastructure across all 7 imaging centers and cores within the university. Liang joined us at Janelia in 2014 as our Director of Advanced Imaging Center, which he will talk more about in his presentation today. So with that, I'm going to turn it over to our first speaker, Kristin Branson.
All right. Thank you for the very nice introduction, Janine. And thank you. I echo Ron's appreciation for you organizing this series. I know it's a lot of work. Okay, so today I'm very happy to tell you about our lab's work on analyzing animal behavior using machine vision and learning. Before I do that, I just wanted to see if this will go. There we go. I wanted to tell you a little bit about the computation and theory area at Janelia that I'm a part of. So computation and theory is a collection of soon to be 10 labs working to build a quantitative understanding of biology and interpret data at scale. One thing that's been pretty unique about Janelia is the recognition that computation and theory are becoming ever more important parts of biology. As I'm sure you're all aware, in the life sciences, our data sets are growing in size such that efficient analyses are necessary automated analyses, are necessary to process the data. And the systems that we're studying are growing in complexity, meaning theory and mathematical abstraction are necessary to interpret and understand them. The two biology research areas at Janelia exemplify this. The first one, which has been going on for a couple of years now, mechanistic cognitive neuroscience. The goal here is to be able to understand how the complex cognitive behaviors are performed at the mechanistic level. To do that, we need to be able to cross scales from kind of the minuscule molecules to cells, to circuits, to populations of neurons, and then finally to the emergent phenomena of behavior that comes out of this. There's been a new research area at Janelia that was announced this week, and you can learn about it on the Janelia website, which is 4D physiology. And that's going to have several of the same challenges where we want to understand tissue at the molecular and cellular levels. And what's been really unique about Janelia is that we collaborate and jointly consider the theoretical principles and the quantitative models that we want to investigate during the experiment design, so very early in the experiment. And we also think about what cutting-edge computational methods can enable before we collect the data. So I'm very happy to have been part of the computation and theory research at Janelia for the past decade. Okay, so I'm going to switch now to telling you about the work that our lab does. And so what our lab works on, as I mentioned in the title slide, is analyzing animal behavior. And we do this from video. And so there's a couple of reasons that this is, I think, an important biology topic. So the first is that these quantitative analyses of behavior are a useful tool in pretty much any field of biology that involves behaving animals. And it's particularly important in neuroscience because behavior is the output of the nervous system. And to understand how the nervous system is functioning and what it's doing, you really need to understand the behavior that it's producing. So there are a couple of types of experiments that we and our collaborators at Janelia work on to using quantitative analysis of behavior. So the first, we are recording neural activity in awake and behaving animals, such as this mouse here, using either electrophysiology or functional imaging. And we're also recording video of the animal behaving. This video is not playing, but this is an example of that kind of video. And our goal here is to be able to understand how the neural activity, what computations the neural activity is performing to produce these behaviors, so to understand those relationships. The other type of experiment that we've spent quite a lot of our own labs working on is experiments where we're able to manipulate the neural activity in the brain using genetic techniques such as optogenetics. So what I'm showing in this top image here is an image of a fly brain. And the neurons that we're genetically targeting are those that are shown in green. These are output neurons from the optic system of the fly. And here I'm showing the behavioral effects of activating those neurons. If you look at this bottom left corner, it'll turn on. And when that light turns on, that means we're activating those neurons. And if you are a connoisseur of fly behavior, you might be able to tell that when we activate these neurons, and it'll play again in a second, all of the flies start walking backwards. And so we'd like to be able to understand these types of changes quantitatively and infer that the neurons that we're activating here are involved in the control of backward locomotion. So quantitative analysis of behavior is a very useful tool, but actually to me, I think it's a fundamental question in biology. So simple animals like the fly, which have really minuscule brains, can efficiently solve problems that are outside of what the most complex AI systems can solve. They can survive, thrive, and reproduce in the wild. They can find food and mates, they can avoid predation, and they can learn and adapt to new environments. And we want to understand the algorithms that animals are using to do this. To be able to do that, we need to know what the vocabulary of behavior is and what the rules of behavior are. So that's what our lab is endeavoring to discover. So we do this using video, as I mentioned before. So video is a really great way to collect information about behavior. You get a lot of information. It's non-invasive, it's relatively inexpensive and easy to collect. And for us whose visual systems are evolved to understand these kinds of images, we feel like we can interpret what's going on in these images relatively easily. However, from a quantitative perspective, video is actually quite hard to interpret. So this image is 1,000 by 1,000 pixels, which means that this video is a 1,000,000 dimensional time series. And so it's quite a high dimensional, high data problem. And we want to be able, what our lab works on is trying to find ways of turning this really high dimensional signal into something that is low dimensional, interpretable, and biologically meaningful description of the behavior that's actually happening here. So the two questions we think about are, what should this representation be? And then second, from the, once we know what this representation is, how should we automate its computation from the video? Okay, so how should we represent animal behavior? One of the dichotomies that we face is whether to use a discrete categorical representation. For instance, we can say that there are 3 behaviors that a fly can do. It can walk, it can stop, and it can turn. Or we could instead use a continuous representation of behavior, for instance, characterizing the speed and distance to another fly. If we choose to use a discrete representation, where do we get these different categories? How do we know that these are the 3 things that flies can do? One thing we could do is we could ask biologists who've been studying animal behavior for centuries for these behavior categories. So in this case, we can encode their intuition about behavior using supervised machine learning, and we can train classifiers that can automate this behavior categorization. On the other side of the dichotomy, we can use unsupervised learning. And so to use unsupervised learning, what we need to be able to do is define mathematically what it means for 2 behaviors to be similar. For instance, we could say that 2 behaviors are similar if the animal's speed and distance to another animal are similar. Once we have that definition of this kind of mathematical definition of what it means for 2 behaviors to be the same, then we can use unsupervised learning methods such as clustering to discover behavior categories. So I thought today what I'd do is overview some of the techniques that we've been developing in both of these 2 areas of supervised and unsupervised machine learning. So in the kind of supervised techniques that we develop, we draw heavily on existing machine vision and learning techniques that have primarily been applied to humans, images of humans, for instance, and haven't been applied to biology. And so we spend a lot of our time thinking about how to reformulate the problems that are important to biologists and get these types of algorithms to work well and solve the right problems for biology applications. And so I'll tell you a little bit about that. And then the second topic that I will tell you about is some very early work that we're doing right now on unsupervised machine learning. And here, there isn't as much machine learning existing working approaches to draw on. So we're really trying to work on developing new algorithms in this case. Okay, so I thought I'd start by telling you about some of the work on behavior categorization that we've done. And so in behavior categorization, what the goal is to assign for each fly and each frame labels of what it's doing at every time point. And so for this fly that's highlighted with a triangle, you can see the automated classification of what our classifiers think the fly is doing at every time point. So we've developed some software called Java to allow biologists to interactively train behavior classifiers. And you can download the code here if you want to try it. So this is a screencast of me training a wing grooming classifier. And so what I'm going to do is I'm going to manually label when the fly is grooming its wings and when it's not. This timeline right here is showing the manual labels. And so you can kind of see what it looks like when a fly grooms its wings. You can kind of see the wing fold underneath the fly. So I'm going to label some frames where the fly is grooming its wings. And then I'm going to label some frames where the fly is not grooming its wings. And notice that I left this little bit of a gap here. And that's because it's kind of hard to tell exactly when the fly stops grooming its wings. So we spent a lot of time on the user interface. And so you can hit this train button. That's the thing I'm going to do next. And the machine learning occurs quite quickly. So it takes kind of 30 seconds to 60 seconds to train a classifier. And after the classifier is done training, the predictions of that classifier are going to appear on this bottom timeline. And so the idea with Java is that it's this interactive training setup where you go through and you look at the predictions of the classifier and you find frames where the classifier is predicting incorrectly the category of the behavior. So here it's predicting that the fly is grooming its wings when the fly is stopped. So maybe from those two sets of labels that we gave it, you know, that the classifier has learned that the fly needs to be stopped, but it hasn't learned that its wings need to be moving. So we can correct the labels here. We can add some new labels to tell it that the fly is not grooming its wings here, and we can retrain. And the classifier works better after retraining. So we iteratively use this kind of process, and it's called active learning in machine learning. Okay, so as we developed methods to categorize behavior, we learned a few lessons that guided our development. So the first thing that we figured out is that we don't know what all of the behavior categories are, and we want biologists to be able to define their own. And biologists disagree to some extent on what the definitions of these behaviors are. So it was really important for us to be able to put this machine learning software in the hands of biologists and let them train their own classifiers. So the second thing that we noticed or we learned is that the kind of standard way that people had been labeling data for training behavior classifiers didn't work particularly well. So what people had been doing in the past is they had taken a video and then they had labeled every single frame in that video. And that doesn't work because some behaviors are much more rare than other behaviors. And so to be able to get enough labels of the right kind of these rare behaviors, you need to label a lot of video. And so we weren't getting big enough data sets of these rare labels. And this is a kind of a now recognized problem in computer vision of these kind of long-tailed distributions of behavior categories. Another thing that we noticed is that sometimes the biologists are unsure of what the right label for the behavior category is. And they would spend all of their time looking at that one frame and agonizing over what the correct label was. And then in the end, actually label inconsistently. So the information that they gave us wasn't particularly useful. And the final thing that we noticed, and so the way that we got around that is we only asked biologists to label the frames where they knew what the right label was. And then the final thing that we decided is that we needed to have a one versus all labeling, meaning that multiple behaviors can happen at the same time. And we didn't want to force that exclusivity of behaviors. Okay, and the last thing that we found quite useful is this interactive training. And that's because data sets are very non-homogeneous, and it's very hard to come up with a training data set that is representative of all of the different ways that you might want to use your classifier. And the important thing to know about machine learning is that the basic assumption is that your training data set follows the same distribution as your test data set. And that's often not the case in real world applications. So to give you an example of that, the first test that we did with Java was this neural activation screen that we performed. So in this screen, we went through a collection of 2,200 GAL4 lines, so different genotypes of flies that each allowed us to manipulate the neural activity in a different sparse subset of neurons. And that's what I'm showing here, images of the brains of these different of flies from these different genotypes, showing which neurons we were activating. The behavioral effects of these activations were quite diverse. So for instance, activating these neurons produced a large amount of courtship between flies, and activating these neurons produced a large amount of jumping in these flies. And if we only trained, say, on our wild type or our genetic control of flies, we wouldn't see these behaviors very often. And so our behavior classifiers wouldn't work very well. We would be kind of generalizing to something we'd never seen. And machine learning does kind of crazy things when you do that. And so it's very important to have this kind of interactive framework where we would train a classifier, predict on a large amount of data, review the results, and then add more labels and retrain. So I'll switch a little bit to talk about some more recent work that we've been doing with kind of similar ideas. So we've been working on something called the animal heart tracker. And the goal here is to be able to predict the pose of the animal. And what that means is with a biologist identifies a bunch of different landmark points that they are interested in knowing the position of in every time point. And we want to know where those landmark points are in every time point. So for instance, it might be the tips of the legs or something that you're interested in tracking. So we've been developing, again, these interactive software to make it really easy to label the types of data sets that are important for training good trackers. So this is a screencast of a senior scientist in our lab, Alice Roby, training a fly pose tracker. So as you can see, she was labeling these different landmark points. We've made it so that you can navigate around in time to look at the temporal context of that frame, which we found to be very important. You can also, as she's doing right now, switch to different videos to be able to get a diverse set of training data. You can switch to different flies to again, get more types of training data. We've also put the training in the hands of the biologist. And with these kind of modern deep learning algorithms, there's a lot of hyperparameters involved. And to try to make that something that biologists can use and understand, we've made visualizations of how these different hyperparameters might be affecting training. And in particular, the ones that, we've also identified the ones that are quite important for different data sets. So you can, as Alice just did, you can hit the train button, within this interface and visualize with this monitor the progress of that training. So deep learning techniques, which these are based on, use something called gradient descent, which is an iterative process for training your classifier. And so it's getting better and better over time. And what you can do is you can see, a quantitative sense of how well it's doing, as well as you can actually track using intermediate versions of that tracker to see how well things are going. And so that's what Alice is doing right now. And so you can hit that track button, it will track, and you can visualize how well things are working and perhaps find new labels to add to the training data. Okay, so just to kind of highlight some of the decisions that we made while we were developing this software. So pose tracking is an active field of research. There are thousands of computer vision papers about pose tracking, and we've designed our system with this in mind. So the exact architecture and training algorithm is just one of the modules that's part of our system. So we've implemented our own training algorithms, but also very popular algorithms. So Deep Lab Cut is very popular for animal tracking, and Open Pose is very popular for human pose tracking. And it's easily extendable. So we've set up our the way that we've written the code to kind of compartmentalize into modules, different pieces. And in particular, we've taken the piece that's relevant, that's very specific to the types of data and the types of interface that we work with, these animal tracking and video, into one piece of code. And that's something that you can call very easily from kind of any TensorFlow and now any PyTorch code to grab a training data batch. So it's quite easy to take our software and incorporate new training algorithms. The other thing that we've done is, as many of you are probably aware, these deep learning algorithms require special hardware to be able to run. They require quite powerful GPUs. And so there are many ways that you can get access to those GPUs. You might want to buy one and put it into your own computer, or actually to be able to have access to a lot of GPUs on demand, you can use the cloud. So for instance, the Amazon cloud, or at Janelia, we've built a GPU cluster here. And so we have all of these different backends that we've instantiated and made available. So when you run APT, you choose a tracking algorithm and you choose a backend to run on to make this quite modular. We've also made this work on a bunch of different types of setups that we've, with our biologist collaborators at Janelia, found to be common. So we can track multiple flies from a single view, or we can track a single animal from multiple views so that we can get 3D tracking information. We spent a lot of time on the quantification of how well these algorithms are working, and this is showing the performance here of the best performing algorithm. What these circles are indicating, so that tiny little red circle is the median error, and the dark blue circle is the 97th percentile of error. So so kind of most errors are well within this blue circle, and half of the errors are within that red circle. So these tracking algorithms are working pretty well, and well enough to be able to do useful biology. But is this animal pose tracking problem solved? And I would say that it's not. So what this plot is showing is the performance of the tracking algorithm and so higher is better, as a function of the number of training algorithms, or sorry, the number of training examples that we give the system. And kind of, there's this question of maybe we just need to label more and more training data, and eventually we'll perform well. This dashed gray line is how well a human can annotate. So we asked our human annotator, a biologist, to annotate the same data twice. And we can see that all of the algorithms are performing much worse than the human annotator. So why is this? So hopefully this video will play. So if you are trying to annotate what's going on in this frame here, it can be quite hard from this single frame. And that's actually what our machine learning algorithms are doing. They're not using video the way that the human biologist is using video. They're not kind of using that kind of temporal context. Instead, the algorithm is just looking at this single frame and trying to decide which of these legs belongs to which fly. So if you have video, all right, still not playing. Okay, so if you have video, you can get temporal context, and sorry, it's not playing, but you should believe me that it's much easier to annotate the positions of these landmarks with temporal context. And so that's something that we're working on, is being able to incorporate temporal context into these kinds of machine learning algorithms. Okay, so I'm going to switch gears now and tell you a little bit about some of the unsupervised learning approaches that we've been trying to discover structure of behavior. So in the past, unsupervised learning, and particularly as applied to behavior analysis, has primarily consisted of either clustering or dimensionality reduction techniques. And as we were talking about in the very beginning of the introduction, this requires us to have a good measure of how similar two behaviors are. If that kind of initial representation of your data is incorrect, then these types of approaches won't work particularly well. And so what people end up doing is they engineer representations based on their own intuitions of what a good representation of behavior is. And then they apply these clustering or dimensionality reduction techniques. And that's actually how computer vision used to be done back when I started doing computer vision back in 2000. People would come up with clever representations for their data and then train relatively simple classifiers to be able to perform the task at hand. So deep learning has really been the thing that's made you have heard about machine learning, right? 20 years ago, there weren't a lot of people talking about machine learning, but now machine learning is a very popular field. And that's because deep learning has made things work much better in practice. And the way that it's been able to do this is that instead of engineering by hand clever feature representations, we start with a raw representation of the data. We start with the pixel representation of images, and we learn a very complicated function as our classifier. And this function that we're learning consists of a bunch of different layers. That's why it's called deep. And each of these layers is a better and better representation of the data, more tuned to the final task at hand. So can we do something like this with unsupervised learning? And that's what we've been thinking about. So there's a kind of part of unsupervised learning called self-supervised learning, where we're taking an unsupervised task. We're taking data that doesn't have labels from a human, but we're using supervised machine learning techniques to be able to do something with that. So in particular, we're looking at behavior forecasting. And here the goal is to be able to take the current environment of a fly. So let's say this fly represented by this red triangle, its current environment and whatever it's been doing in the past, and predict what it's going to do in the future. And so if we can come up with a good machine learning model of of predicting the future, the idea is that we've learned something very fundamental about animal behavior. It's a non-trivial task to be able to predict what a fly will do in the future. So we've been applying this type of approach to data that looks like this. We have video of 20 flies, 10 male flies and 10 female flies interacting with each other and exploring a relatively sparse environment. Okay, so why would we want to be able to do this? Why do we want to predict what flies will do in the future? So if we train a machine learning model that works exactly like an animal does, then understanding animal behavior reduces to understanding this machine learning model. So there's kind of two big questions there that we need to be able to answer to do this. So first, does the machine learning model work like the animal? Is it even worth analyzing? And second, machine learning models are actually quite complex and difficult to interpret, that we don't have great ways of interpreting these machine learning models. So how do we interpret the machine learning model? So, there's kind of this risky question: have we just traded one really difficult question of, understanding animal behavior to two really difficult questions of, does the animal work like the machine learning model, and how do we interpret the machine learning model? However, we feel like this is a risky but potentially rewarding direction to explore, and so we've been working on this. And so we've been training a type of machine learning classifier called a recurrent neural network to be able to predict the behavior of flies. So we take in as input the current environment of the fly, as well as a learned representation of the past of the animal. And that's something that I really like about these recurrent neural networks, that we're learning a representation of the past of the animal that encompasses everything that's important about the past behavior of the animal and predicting what the fly will do in the short-term future. So we can take that predictor of what the fly is going to do in the future, and we can actually use that to simulate fly behavior. So we can take the predictions from a given frame, we can apply those predictions, we can create a new environment based on those predictions, and we can predict again. So we can do that many, many times for many flies in a dish, and this is what that looks like. So this is a video of all simulated flies, and probably most of you are not used to watching flies in a dish for which I've been doing for the last 15 years of my life. But it looks pretty good. And just to kind of give you a test of that. So these are 4 videos. One of them is real flies, and the other 3 are simulations from different types of classifiers. And one thing we were trying to do as we did this was to figure out which type of machine learning model was worked the best. So the question is, which of these 4 is real flies? Okay, so the answer is number 2 in this top right corner, and the 3 different models that we're trying are shown here. Okay, so this is still very much work in progress. So we're still kind of in this process of trying to understand, does this machine learning model work like the animal? And then the next big question is kind of this question of interpretable machine learning or explainable AI. And that's kind of an emerging field in machine learning. And we're hopeful to kind of take approaches from there and develop our own approaches to apply here. Okay, so I'm going to end my presentation with just a short plug for the computation and theory research area at Janelia. And just to, as I told you a little bit about it in the beginning, but I just, wanted to let you all know that a lot of the labs have open positions for postdocs or programmers or postbacks. And in particular, I wanted to let you know about the Janelia Theory Fellow Program, which is an independent postdoc position that we're seeking applications for. And for all of these positions, we really value diversity, and I'd like to particularly encourage members of underrepresented groups to contact me if they are interested in a position. All right, I'm going to hopefully end there with thanking my lab members who did all of this work and our collaborators, and thank you for your attention.
Thank you, Kristin. That was a great talk. I admittedly did fail the eyeball test. So I want to tell the audience that if you have any questions for Kristin, please go ahead and write them in the Q&A box. I'm going to try to get to as many people as possible. And I will call on you to ask the questions yourself unless you submit it anonymously, and I'll ask Kristin. So I want to start with this question, Kristin. With the appropriate imaging and recording platform, are there any constraints in the types of animals or the number of animals or the number of parts that can be analyzed with this software?
Not that we know of yet. I mean, I think, it's the proof is in the pudding, right? We need to test it to make sure that it would work, but we've tried to develop things to be as general purpose as possible. And kind of our rule of thumb is that if it's a relatively easy problem for a human to solve, then we should be able to make the computer vision able to solve that problem.
Great. So actually, this sort of goes nicely with some of the attendee questions. So one attendee wants to know, would it be possible to use this software to study plant growth, which would mean analysis of large photo libraries? What do you think about that?
So I don't really know what the questions involved in plant growth are. We do have, my former labs have actually looked at kind of plant identification. Certainly, yeah, there's no reason that this has to be an animal that you're doing, part tracking on or behavior classification tracking or behavior classification on. You just need to have some kind of data that's input to the computer. But it would, yeah, I'd love to hear more about kind of what the types of problems we actually want to solve there are to give a better answer there.
And how about, this is a little bit closer to home, how about the possibility of using it for zebrafish behavioral analysis?
Definitely, yeah. We haven't done it just because, we just haven't done it. But yeah, I think it would be, there's no reason to think that it wouldn't work. And yeah, hopefully we will start working at some point with the RN's lab at Janelia on that kind of a problem. And yeah.
So another audience member wants to know, is it possible for some of these algorithms to take into account various depth fields of view from a camera? For example, distinguishing a mouse grasping a pellet right next to the camera or far away from the camera?
Yeah, so the way that we've done this so far is to collect multiple videos from multiple cameras and then triangulate to get 3D information. I think there is some amount of work in computer vision where people are trying to estimate 3D from just 2D information, right? So when we watch TV or something like that, we're not totally lost on kind of the depth information of what's happening in that video, right? So we don't have kind of any kind of stereo information when we are watching TV, but we're able to estimate depth approximately. And so people are using that kind of technique and able to get some kind of depth information out. The kind of question is whether that's accurate enough to do the type of biology that we want to do. Like I'm not confident that it would be good enough to understand, kinematics of or biomechanics in animals. You might need more precision, in which case it really would be important to be able to have multiple cameras. And so we're kind of thinking about both approaches right now. We have, we have a couple, 2 camera systems set up, but to be able to do the types of things we want, we would probably need, 10 cameras, 20 cameras to get kind of a big field of view for a fly to walk around in, say. And we're also thinking about, how much can we do from, just one camera if we have a really good model of a fly, say.
Thank you. We also have a question from an audience member, Aman. Aman, if you're there, you can unmute yourself and ask your question.
Yeah. Am I audible?
Yep.
Yeah. Hi, Christine. This is Aman from NCBS Bangalore. I do quite a bit of flight-based tracking and AI analysis. I've developed my own algorithms. So, but there, as you mentioned, that mostly they are based on single frame analysis. So that's what I wanted to ask, like, are you implementing the RCNN or LSTM based models for pose estimations or predictions of the action predictions? And if yes,
yeah,
so what are the challenges?
Yeah, I mean, it's a thing that we're thinking a lot about. So we haven't implemented them yet. It's kind of work in progress right now. And we are trying both of these. We've done some behavior classification stuff with both convolutional over time networks and with recurrent neural networks. And I think the main challenge, in my opinion, is going to be the amount of temporal context that you need. So these kind of it might be that you need hundreds of frames of information, say, to be able to really disambiguate what's going on in a given frame, because the animal might just sit still in that ambiguous position for a long time. And so the kind of current algorithms They require you to kind of put a lot of information on the GPU at a time if you want to have a long temporal window that you're looking at. And so those, we're kind of, the kind of time window that you can look at is pretty limited with RNNs or with temporal convolutional networks. And so we're thinking about how we can get longer time windows by having something like a salience detection of when things are changing, for instance. But we don't have any concrete ideas yet. That's just kind of things we're thinking about.
Kristin, one last question before we move on to Liang's talk. This comes from Banapa Unger. Do you think these algorithms would work for either time-lapse video or 3D video?
Yeah, I think it would be interesting to try. There's no reason that they wouldn't be able to, I would say. That's the kind of nice thing about machine learning is it can try to learn to adapt to any kind of setup as long as you have enough training data and as long as the information is there.
Great, thank you. I think that's about it for time right now, and we're going to go ahead and move on to Teng Liang Chu.